My name is Jose Burgos Portugal, and hello and good afternoon to everyone. And I'm here today to talk to you about a very fascinating research topic that we work at the Mitchell Lab called autophagy hijack, the tactic used by Campylobacter concisus to survive in your gut. So what is Campylobacter concisus and what are its associated diseases? Well, Campylobacter concisus is a gram-negative curved spiral rod. It is motile by a single polar flagellum. It is considered an emerging gastrointestinal pathogen that may be associated with a range of diseases. So if one of those first associations is acute gastroenteritis, which causes you to have severe debilitating pain in your stomach and basically causes you to have really bad diarrhea, which is really unpleasant, really. There's also a second association with inflammatory bowel disease, and as Vicky mentioned earlier, it is a chronic lifelong disease that can basically be subdivided into two main subtypes, ulcerative colitis and Crohn's disease. Now, ulcerative colitis causes ulcerations in the colon, as you can see here on this image on the left, while Crohn's disease causes mass inflammation throughout the gastrointestinal tract, but I've shown here as an example an inflammation in the ileum. A third association is Barrett's esophagus. Now that is caused by gastroesophageal reflux from the stomach right through to the lower esophagus. And this causes basically a change in the cellular lining of this lower esophagus, as you can see here in this image on the right. And this is termed a metaplasia. And this essentially becomes a predisposition to a type of cancer called esophageal adenocarcinoma. However, we've also shown that Campylobacter concisus has also been found in healthy individuals, and this has led to much debate about its role as an intestinal pathogen. However, we cannot neglect the fact that C. concisus has a high level of genetic heterogeneity, by which I mean genetic diversity. For instance, 4 genomic species, which basically means different appearance traits variants within one species, has been proposed. And we've also shown that there are phylogenetic differences, that means evolutionary differences, amongst Campylobacter concisus strains. And we've just shown, particularly the ones we work with in our lab. And this has been shown here on a 16S RNA tree. And what's really interesting is we've shown that this variation is actually being classified into 3 different disease states. Showed very interesting results. Now, it is this level of genetic diversity and also phylogenetic diversity that allows Campylobacter concisus to have a very wide and diverse pathogenic potential. And this is clearly reflected on this table here, which basically denotes results done in vitro using a Caco2 cell line model, showing both the invasion and adherence results. As you can see here, there is an apparent difference in the pathogenic potential amongst the Campylobacter concisus strains isolated from more of a chronic disease state, which is the first 4 here, as opposed to more of an acute strain and as well as a healthy control, which show really low invasive levels. In terms of the attachment potential, the chronic strains again show very high potential to attach. But the healthy control in the acute gastroenteritis strains show very variable results from low to high. Now, we've interestingly found a plasmid within a particular high invasive strain called Campylobacter concisus UNSW CD. That's been shown here. And more interesting of all, we've shown this plasmid to be present from patients with chronic intestinal disease. And this raises a potential question. Does this plasmid confer any invasive potential with these strains? So we know that bacteria can actually adhere to and also invade these cell lines, but we don't know exactly what happens after the bacteria invades host intestinal cells. And this essentially becomes a key research question which I'm trying to investigate throughout my PhD. So this is where also processes like autophagy, which my title of my presentation represents, come into play. And autophagy essentially means a self-eating process. And I can use a simple analogy to explain that to you all. I'm sure all of you have either heard or played the Pac-Man arcade game. And autophagy also shows works in a similar fashion, whereby in Pac-Man you have where Pac-Man eats up all the ghosts in their game. In autophagy, you also have that process occurring, also where the autophagy is actually eating up any foreign matter or molecules that are causing us more harm than good. So normally, an intestinal pathogen would invade the cell and either form a vacuole of some sort or capsule to protect itself once inside the cell. And this can then interact with the autophagic process, and this can occur through the autophagosome. The autophagosome can then fuse with a lysosome, and this lysosome complex actually forms an autophagal lysosome, and this actually causes the bacterial clearance of the cell in the system. And voila, the bacteria is gone. But some bacteria over time have evolved to actually manipulate autophagy to reside and evade it. So an intestinal pathogen again will interact with the cell and fuse with the lysosome, and at the level either of the autophagal lysosome or at the autophagosome, you actually have manipulation of autophagy, allowing the bacteria to persist and stay within the cell to cause further infection. And this, some examples of pathogens that do this are Yersinia paratuberculosis and Legionella pneumophila. Now, this raises very interesting questions with my research project in terms of how important is autophagy to Campylobacter concisus' pathogenicity, and does C. concisus also have a similar ability to manipulate the system like other intestinal pathogens do? So if we recall, I showed on the previous table 8 different strains, but to carry on this investigation, we chose 4 particular strains. UNSW-CD, which showed a really high invasion potential, and also 3 acute gastroenteritis strains, which showed low invasive potential. Now, in order to modulate autophagy inside these CACO2 cells, we chose 3 particular chemical compounds to do so. The first one was rapamycin, which targets mTOR to positively induce autophagy. The second one was 3-methyladenine, which targets class 3 phosphatidylinositol-3 kinase to basically inhibit autophagy. And the third one was chloroquine diphosphate, which targets lysosomal pH levels to positively regulate autophagy, autophagosome formations. So what do we essentially find, and what progress have I made thus far from the commencement of my research project? Well, basically, we found that irrespective of whether the C. concisus strain was derived from a chronic or acute patient, it showed very significant invasion results upon addition of 3-methyladenine. We're recalling that 3-methyladenine here acts as an autophagy inducer. And this was in opposed to no treatment with 3-methyladenine, which caused some invasion with UNSW-CD or no invasion with the acute gastroenteritis strains. So we showed, if I focus on UNSW-CD here, that we showed a significant increase in the invasion levels upon 3-methyladenine addition, as I've mentioned. Chloroquine diphosphate caused bacterial clearance of the inside CACO2 cells. Rapamycin caused very low invasion levels of C. concisus UNSW-CD. And upon looking at the acute gastroenteritis strains, we've shown a very significant increase in the invasion levels as opposed with 3-methyladenine treatment as opposed to without. Next, we wanted to look at how the process of autophagy is occurring in CACO2 cells. And we chose to focus here on the autophagosomes directly, which are stained in red on panel A. And basically, panel A shows you how ubiquitous this process is occurring within CACO2 cells. So the autophagosomes here are occurring around the periphery region. Panel B shows you that there is an interplay between the process of autophagy and the bacteria. Bacteria here are labeled in green, residing inside the cell. So there is definitely a process happening. Panel C, on the other hand, shows you that there is an interaction between the autophagosomes and the bacteria themselves. This is termed a co-localization effect. And these are shown in the yellow regions. Now, in order to determine the internalization of Campylobacter concisus UNSW-CD inside CACO2 cells, we chose to use a transmission electron microscopy approach. The first panel shows you that the bacteria are actually residing within the CACO2 cells, and very novel findings to do so. Panel B shows you that the bacteria shown in the arrow here are actually in close proximity to the autophagosomes or in close interaction, and the autophagosomes here are actually shown in this circular region. Panel C, on the other hand, shows you that the bacteria, C. concisus UNSW-CD, is actually residing within the autophagal lysosomes. And this is actually a very important finding because normally bacteria will be degraded in this complex. So the fact that we're seeing the bacteria here shows you that there could be a manipulation process happening. And this is actually reflected in the results that we actually showed looking at the autophagy gene expression levels upon addition of infection of Campylobacter concisus UNSW-CD in CACO2 cells. So we showed here that there was an actual inhibitory and a dampening effect by the bacteria, which actually shows some exertion of manipulation throughout the process, and that's occurring very widely from the initiation to the bacterial clearance stage. So we've shown here, through the results of the upregulation of MAP-LC3B, which normally is a marker of autophagosomes, the upregulation is telling us that there is an actually inhibitory process happening at this level. And this is actually, in retrospect, reflects the results shown in the direct regulation of these other 4 genes that we've seen, ATG4B, LAMP1, and the cathepsins S and D, which actually act on the phagosome and lysosome fusion process and the bacterial clearance processes, respectively. So to conclude, some Campylobacter concisus strains are capable of manipulating autophagy, and ultimately higher intracellular levels are more likely due to the evasion of the autophagy process rather than the increased invasive ability of each Campylobacter concisus strain themselves. So further research that I would like to do will initiate use of further investigation into autophagy with other Campylobacter concisus strains, and to further that research and follow that up with in vitro studies to look at the intracellular survival levels once inside CACO2 cells for an extended period of time. Now, if you recall, I had very interesting results with the acute gastroenteritis in concisus strains upon addition of 3MA, so it'll be nice to really follow that up. And future directions will concentrate more on in vivo mouse model studies using both an autophagy model and an inflammatory bowel disease model to carry out more functional studies with using those mouse models. And this leads me to my final conclusion and wrapping this up. Just to thank my lab mates for their awesome work and support, our funding bodies, and our collaborators. And